All right, well, good afternoon, everyone. Welcome to Department of Medicine Grand Rounds. Just want to take you through a few things on this slide. If you could put your Zoom window in full screen mode, that's probably the best way to see this event. If you have questions, type them in the Q&A box. We're going to have a couple great presentations and then time for questions at the end. Closed captioning is available at the bottom of your screen, and then I was asked to announce that the session will, of course, be recorded and then posted on YouTube. It will actually be on the Department of Medicine YouTube channel now, no longer the School of Medicine YouTube channel, and you can see the link that's been provided there. So with that, let's get started. So for those of you who don't know, I'm not Bob Wachter. I'm Andy Josephson, the chair of the Department of Neurology. Bob's asked me to step in today and host this Department of Medicine Grand Rounds. Bob is fine. He's taking some time off. I don't really know where he is, but by understanding, he's either golfing or tweeting or golfing and tweeting, one or the other. So we hope Bob's getting some time off and will rejoin us for the next Grand Rounds. Today, we're going to deal with a really exciting topic. So as many of you know, in the setting of the pandemic, there's been a lot of non-pandemic medical news, but in the world of neurology and geriatrics and primary care, there's probably been nothing more interesting than the tale of Aducanumab, the first FDA-approved therapy for Alzheimer's disease in decades, but one that has been met with a lot of controversy, discussion, and today we're going to tackle those issues. Where are we with this drug? Where are we going with this drug, and what should we do practically? We have two great speakers lined up for you. I'll introduce them one at a time. And then again, please, as we go, put your questions in the Q&A, and we'll have plenty of time for questions at the end. So our first speaker is from my department, Dr. Gil Rabinovici. Gil is the Edward Fine and Pearl Landreth Distinguished Professor of Neurology here at UCSF. He's also has secondary appointments in the departments of radiology and biomedical engineering. He did his training here at UCSF after medical school at Northwestern and undergrad at Stanford. He is an expert in caring for neurodegenerative diseases. His work really looks at how structural, functional, and molecular brain imaging techniques can more accurately diagnose conditions such as Alzheimer's disease. He is the co-director of our NIH-funded UCSF Alzheimer's Disease Research Center, and he chairs a large study, the Imaging Dementia Evidence for Amyloid Scanning, the IDEAS study, and the New IDEAS study, as well as being a PI on several national and local studies that look at, again, amyloid imaging and other ways that we can more accurately diagnose early Alzheimer's disease. Gil has won countless awards that I won't go through right now. Relevant to this topic, Gil recently wrote, I think, a very nice balanced piece on Aducanumab that was published in the New England Journal of Medicine. And today, Gil's going to join us to take us through some of the evidence regarding Aducanumab. Gil, thanks, and welcome to the Department of Medicine Grand Rounds.
Thanks so much, Andy. It's really a pleasure to be here and to be presenting with Dr. Redberg. Can you see my slides?
You're good.
Okay, so let's just start with some important disclosures. UCSF Memory and Aging Center was an enrollment site for clinical trials for the drug that we're going to be discussing, Aducanumab, though I wasn't personally involved in these trials, but I have done some consulting work for Eisai, which has a commercial interest in this drug, and you can see my additional disclosures. And then in putting together this talk, I was in the interesting position of trying to get data about a study that hasn't yet been published. Even though the results of Aducanumab's phase 3 studies have been in the public domain for about 2 years, there is still not a peer-reviewed publication. And so the source of the data slides that I'll be showing come from two publicly available locations. One is the presentation by the sponsor at a scientific conference, the Clinical Trials in AD conference, and the other is the materials that were presented for the FDA Advisory Committee that deliberated about this drug. And so just to set the context for talking about a new therapy for Alzheimer's disease, there's absolutely no doubt that Alzheimer's represents an urgent public health emergency. These graphs from the Alzheimer's Association Facts and Figures that is published annually shows that currently it's estimated that about 6 million Americans have Alzheimer's disease. And you can see stratified by age, the expected increase in prevalence over the coming decades. So really more than a doubling in the prevalence of the disease that's driven largely by an aging population. Just thinking about lifetime risk at age 65, the disease will affect 1 in 5 females, 1 in 9 males. Blacks and Latinx are at higher risk than non-Latinx whites. And even though we think about AD differently than, say, cancer or cardiovascular disease, this is ultimately a fatal disorder. It's currently considered the 6th leading cause of death in the US, and that's almost certainly an underestimate. And furthermore, this translates, of course, into dramatic public health costs. So it was estimated that about $300 billion were spent on AD care in 2020, and about 2/3 of that bill was footed by Medicare. Now, the neuropathology of Alzheimer's disease for over a century has been defined by 2 brain lesions. So we have extracellular plaques that are composed of aggregated forms of amyloid beta and neurofibrillary tangles that are intraneuronal and composed of hyperphosphorylated tau. And one of the major advances in AD research in the past 15 years is the development of biomarkers, initially in CSF, then PET tracers, and more recently, plasma biomarkers that can detect these key pathologies in living people and give us a window into the evolution of the disease in humans. And so what I'm showing you here on the top row are amyloid PET scans, and on the bottom row are tau PET scans. First, from a cognitively normal person, this individual doesn't have amyloid in their brain. So this red binding that you see here represents just nonspecific white matter binding. But if you look at the medial temporal lobes, you can see early evidence of tangles. And sadly, all of us beginning at about age 50 or so are starting to develop some tangles in our medial temporal lobes. Now, here's another cognitively normal person, but this individual already shows a lot of amyloid throughout their neocortex. And it's really in the presence of this amyloid that the tau tangles start to escape from the medial temporal lobe into the neocortex. And so this brings up an important principle, which is that the pathology of amyloid and tau really precedes clinical symptoms in AD by decades. And this defines a large preclinical phase of the disease and potentially a phase for early intervention. Now, completing the picture, here you can see an individual with mild cognitive impairment and someone suffering from full-blown AD dementia. And these scans bring up some additional important points. So first, amyloid reaches a relative plateau fairly early in the disease. You can see not much difference in amyloid between someone who's cognitively normal and someone with dementia, but it's really in the presence of amyloid that tau spreads from a relatively innocuous process in the medial temporal lobe into the neocortex. And then it's that progressive spread of tau that precedes neurodegeneration and ultimately accompanies cognitive decline. So now zeroing in at the molecular level, the amyloid beta that forms plaques is derived from the amyloid precursor protein, or APP, which is a transmembrane protein that's expressed in neurons. And sequential cleavage of this protein by beta and gamma secretase generates polypeptide fragments. And it's primarily the 42 amino acid version of these polypeptides that starts to aggregate in the extracellular space, initially forming highly toxic A-beta soluble oligomers, and then eventually fibrils and amyloid plaques. And these amyloid aggregates are neurotoxic. They interfere with synaptic function and network function. They trigger what ultimately turns out to be a maladaptive inflammatory response. They promote phosphorylation and aggregation of tau. And so this conceptual model where aggregation of amyloid leads to a downstream cascade of events that ultimately results in synaptic loss and clinical dementia is called the amyloid cascade hypothesis. And this has really been the major focus of drug development in AD for many years. So this finally brings us to aducanumab. Aducanumab is a human Ig1 antibody. As shown in this plot, it binds selectively to A-beta oligomers and fibrils, but not to monomers. Really interestingly, it was found by screening human B-cell libraries that were collected from older adults who showed no or slow cognitive decline as they aged. In preclinical models, it was shown that this antibody can then promote microglia-mediated phagocytosis of amyloid and a dose-dependent reduction in amyloid plaques. And this was initially shown in mouse models of AD, where you can see increasing doses of antibody lead to reduction of plaques as measured by 6E10 antibodies or thioflavin in cortex and hippocampus. And these results were then replicated in a human phase 1b study And the results were so dramatic in terms of the before and after, showing dramatic amyloid reduction, that this made the cover of Nature. We had never seen such a dramatic biomarker response to any AD therapy. And so this generated a tremendous amount of excitement, and actually led the company, Biogen, which is the sponsor of the drug, to move directly from phase 1b to a large international phase 3 program with two identically designed randomized clinical trials called Emerge and Engage. These were large international studies. The primary inclusion criteria included people who had early clinical AD, so either mild cognitive impairment or very mild dementia. And this was intentional because the thinking was, as I showed you from the biology of the disease, that the response to amyloid lowering therapies is more likely in earlier clinical stages of the disease than in advanced disease. The primary criteria included an MMSE range of 24 to 30 and a global clinical dementia rating of 0.5, which means that these individuals were still largely functioning independently despite their cognitive decline. They were randomized equally to receive either low or high dose antibody and placebo. And the primary endpoint was a scale called the Clinical Dementia Rating Sum of Boxes at 18 months. This is a patient-oriented scale. It measures important aspects of cognition and daily function. And then there were a number of secondary endpoints, the MMSE, ADAS-Cog, which is a cognitive measure, and an activities of daily living, a functional scale, as well as sub-studies that looked at amyloid PET, tau PET, and spinal fluid biomarkers. Now, after all that fanfare and excitement, things ended really abruptly on March 21st, 2019, when the sponsor announced that they were terminating both Engage and Emerge because a pre-specified futility analysis that was performed with pooled data from both trials had met the criteria for early study termination. And this was incredibly disappointing, of course, to people who were in the studies and conducting the studies. But to be honest, we've kind of grown used to this narrative in AD, drug that looks very promising early on and then fizzles in phase 3. The interesting plot twist happened about 7 months later on October 22nd, when in a 180-degree turn, the company reported that actually upon reviewing further data, one of the studies had met its clinical endpoint and they were proceeding with a biological license application to the FDA. And since this occurred around the time of Halloween, it raised some interesting comparisons with zombies. And this was a drug that was literally rising from the dead. And so the data that I'll be reviewing today are the data from these early terminated trials. And so you can see at the time these trials were terminated, about 60 or 66% of participants had completed Emerge and Engage respectively. This represented about 10% more data than was available for the futility analysis. The analyses I'll show are modified intention to treat for the full data set. However, all clinical observations after study termination were censored. And I'll also show some results from the amyloid PET and CSF and tau PET substudies. So here are the basic demographics for the studies. The average age was about 70, just over half of the participants were female. There was very poor racial and ethnic diversity in the study. About over 50% of individuals were already on AD medications like cholinesterase inhibitors, and so any drug effects occurred above and beyond the standard of care. About 2/3 of individuals carried the ApoE4 risk allele, which is the most common risk allele for developing sporadic Alzheimer's disease, and about 80% of individuals had mild cognitive impairment, 20% had mild dementia. So this was a very mildly impaired group. So here are the primary endpoint results, change in the CDR sum of boxes. You can see changes in placebo in gray, low dose aducanumab in blue, and high dose in purple. And so increases in this scale mean progressive clinical worsening, so everyone worsened, but you can see that at 78 weeks, people who received high-dose aducanumab worsened a little bit less than those who received placebo, and this was statistically significant. There was no difference in the low-dose group, and so the magnitude of this effect was about 0.4 on this CDR sum of boxes. The placebo group advanced about 1.7 on this scale. And so this translated into about a 22% reduction overall in clinical progression. Now, when you look on the right at the results of the ENGAGE study, there was really absolutely no difference between placebo, low dose, and high dose aducanumab treatment. In fact, numerically, the high dose group did worse throughout all the time points, though this was not statistically significant at any point. And so really divergent results in terms of clinical outcomes. And when you look at secondary cognitive and functional outcomes, the story remains very much consistent but confusing between the two trials. So consistently, we see a slowing of decline on MMSE, ADAS-Cog13, and ADCS-ADL in the EMERGE study in the high dose group compared to placebo, but absolutely no effect for low dose in either study and for high dose in ENGAGE. And so we'll talk a little bit about why these divergent results may have occurred in two identically designed studies in just a couple of slides. But first, let me present some of the biomarker results. So here we look at changes in amyloid PET. You can see placebo in red, low dose aducanumab in blue, and high dose in green. And these results were actually very consistent across both EMERGE and ENGAGE. You can see a fairly dramatic dose-dependent reduction in amyloid plaques as measured by PET scans. This was about a 55 to 65% reduction across the two studies. Similarly, in both studies, in the small subgroups that had CSF collected, there was a treatment effect on phosphorylated tau, which is an AD-specific biomarker of tau phosphorylation, though no effect on total tau measurements. Total tau is thought to be a less specific marker of neurodegeneration. And in a very small number of participants who had tau PET, here you can see some more before and after pictures. There was, and quantification on the right, there was an increase in tau PET signal in the placebo group and either a stabilization or actually a reduction in tau PET in different brain regions in the treated groups. And so some suggestion, though the numbers are small, that reductions of amyloid may actually be affecting downstream biology of tau. Now, the primary safety concern in both these studies were amyloid-related imaging abnormalities, or ARIA. These can occur as either edema, shown here as white matter hyperintensities on flare sequences on the top, or as microhemorrhages or superficial siderosis, hemosiderin deposits at the peel surface of the brain, shown here on hemosiderin sequences of MRI. And these were in fact expected adverse effects related to the drug. They seem to be a class effect of the anti-beta monoclonals. They may have to do with changes in vascular permeability that's related to treatment, or possibly clearance of vascular amyloid directly. They're associated with higher dose of drug and with the ApoE4 genotype. They were very common and pooled across the high dose groups in both studies, occurring in 40% of the drug arm compared to 10% of the placebo arm. Now, about 3/4 of the time, these events were actually asymptomatic and only detected based on surveillance MRI scans. But in 1/4 of the patients, there were symptoms, usually mild and nonspecific, like headache or dizziness, but occasionally severe with focal neurologic deficits. The symptoms and the ARIA-E changes resolve over time with treatment suspension and dose reduction. And actually, the overall discontinuation rate was about 6% in the study. Another important point is that ARIA was much more common in ApoE4 carriers, about 40% in E4 carriers compared to about 20% in non-carriers. Now, there has been a lot of time spent trying to understand the differences between EMERGE and ENGAGE. And this has literally led to a year of FDA deliberations back and forth with the manufacturer. It has led to hundreds of pages of post hoc analyses. I think we can get into this in the Q&A. For the sake of time, I won't describe in detail some of the possibilities that are out there, but suffice it to say that we don't really understand why these trials behaved very differently. There were differences in the conduct of the studies in terms of exposure to high dose drug. There were differences in performance of the placebo group that may be just reflecting the unpredictable heterogeneity of progression in early onset AD, but really all of the explanations are based on post hoc analyses that are fraught with pitfalls. And this has been a major source of consternation and concern that we have these conflicting results. However, the results of aducanumab can also be compared to other drugs in the same class that actually are showing a very similar pattern of both biomarker and clinical results to what was observed in eMERGE, the positive study. And I'm just going to highlight one study, which is donanemab. This is an antibody that targets a different epitope that's specific in amyloid plaques. And I want to highlight this study, which is a phase 2 study that was published earlier this year in the New England Journal, also because it took an interesting and new approach to a couple of elements of study design. So only patients who had intermediate levels of tau as determined by PET were included in the study. The thinking was that people who had very low tau would probably not progress enough in a trial, whereas people who had very high tau may not respond to amyloid lowering. The other interesting thing is that patients were treated based on their PET response. So once the PET scan turned from positive to negative, they were actually switched to placebo. This is kind of similar to induction chemotherapy in that you don't continue the treatment indefinitely. And so here are the results of this study. First, amyloid PET, dramatic and early reduction in amyloid PET signal. Like with aducanumab, this translated into a slowing of progression of tau PET signal, though this was done in a much larger sample than was done in aducanumab. And again, this translated into a modest but statistically significant clinical benefit. And so there's an emerging story here, which is that at an early clinical stage, reducing a lot of amyloid can translate into slowed progression of tau and into modest slowing of clinical decline. And so this is where the FDA advisory committee and the FDA were left in contemplating these results of very complicated data in two studies that had very different clinical results that were complicated by early termination of the studies, by differences in dosing and protocol implementation. And I said at the time, and I will say again, that I think that the most logical next step would have been a third phase 3 study of high-dose drug versus placebo to determine if the results of eMERGE, the positive study, could be replicated. Now, as we'll hear from Dr. Redberg, the FDA took a very different approach, and I think kind of lost in all the controversy around the regulatory process, around the pricing of the drug, is that there is a scientific signal here for benefit. And it's coming not just from aducanumab, but from the other drugs in class like donanemab that I showed you, that we have to acknowledge that the clinical benefit of lowering amyloid is modest at best, but it is very consistent in these latest generation of antibodies. And so whether it turns out to be aducanumab or another one of the drugs that may follow in its footsteps, I do think that we will look back in the future at this time as the beginning of a new era of Alzheimer's care, where we transition towards a biomarker-supported approach to diagnosis and molecular-specific therapies. If you think about other complex diseases like HIV, multiple sclerosis, the first-in-class drugs were never as good as we wanted it to be. The efficacy was lower than we would have hoped for. The toxicity was higher than we liked. And yet these drugs like AZT, for example, and HIV they provided a foothold. They opened the door to better treatments. And in a complex disease like AD, I think it's going to be very important for us in the future also to focus not just on amyloid, but on many other elements of AD pathophysiology, likely with combination therapies that will target different elements of this complex disease. And ultimately, I think those are the approaches that we will need to make a profound impact on disease outcomes. And so I will end there. I'd just like to thank the clinical trials team at the Memory and Aging Center, led by Adam Boxer and Mary Kessler, the leadership of the MAC, Bruce Miller, my lab, collaborators, and funding sources. So thank you very, very much.
All right, thank you, Gil. We've got a lot of people putting in questions in the Q&A, and we'll get to them at the end. And now let's move on to Dr. Rita Redberg, who needs no introduction, but I'll do it anyway. So Rita is a cardiologist, professor of medicine here at UCSF since 1990, and core faculty in the Philip Lee Institute for Health Policy Studies. She grew up in Brooklyn, graduated from Cornell and Penn Medical School, and had a master's at the London School of Economics as a Penn Thorne Fellow. She's the editor-in-chief of JAMA Internal Medicine since 2009 and has spearheaded the journal's focus on healthcare reform and less is more. As you all know, her research interests are in the area of health policy and technology assessment and how to promote high-value care, as well as the need for inclusion of women in clinical trials. She has served a 6-year term on the Medicare Payment Advisory Commission, which advises Congress on Medicare payment issues. She also served on the Medicare Evidence Development and Coverage Advisory Committee. She's given congressional testimony a number of times related to regulatory issues and has worked in the office of Senator Hatch and with the Senate Judiciary Committee on FDA-related matters during her tenure as a Robert Wood Johnson Health Policy Fellow. In addition, as many of you know, Dr. Redberg is now an elected member of the National Academy of Medicine. Rita, thank you so much for being here. Take it away.
Thanks so much, Andy. It's great to be here with you and Gil and talk about this really interesting approval. So I'm going to pick up where Gil left off and talk about the science and the politics, which are really interesting behind this approval. Okay, so these are my disclosures, which you mostly mentioned already, and I have no financial conflicts. Okay, so let's talk about this aducanumab approval, as it had many unusual and troublesome aspects. Firstly, the FDA explicitly told its expert advisory committee that they were not going to consider an accelerated approval for this drug. And the advisory committee voted on the basis of clinical benefit, and Gil has told us about those trials. They were both ended for futility. They did not meet their endpoints. And 10 of the 11 expert members of the advisory committee voted that there was insufficient data to show that aducanumab slows cognitive decline. The 11th was uncertain, so nobody supported any clinical benefit from this drug. And then again, as Gil has mentioned, the FDA surprised many and contradicted itself and approved aducanumab on the basis of a surrogate measure using the accelerated approval pathway, which it had said it would not be considering. And to remind you all, the accelerated approval pathway is meant for diseases that are life-threatening and they have no available treatments, and it does allow for approval based on a validated surrogate measure. And then a post-market study is required, which would confirm clinical benefit. A surrogate measure is a lab measure or an image, and Gil took us through those, that by itself is meaningless to patients. It's a very nice image and picture, but a surrogate marker is not something that helps patients. It's only helpful to patients if that surrogate marker is validated and actually predicts clinical benefit. Unfortunately, the surrogate marker on which aducanumab was approved has not been validated and has not been shown to predict Alzheimer's disease. And in fact, the theory that amyloid plaques cause Alzheimer's remains controversial and unproven. Certainly, it's true that some people with Alzheimer's have amyloid. It's also true that some people with Alzheimer's don't have amyloid. It's also true that some people with amyloid don't have Alzheimer's. And it's possible that it's a neurodegenerative process that happens with aging. Alzheimer's and other dementias increase with aging, and they could be true but unrelated. So it's not a valid surrogate marker on which this approval was based. Additional features that are troublesome about this approval is that Biogen only studied patients who had early Alzheimer's disease, because this was meant to be a preventive therapy. And they also applied many exclusion criteria. For example, they excluded people who had cardiovascular disease, which many older persons do. They excluded people who had who were taking anticoagulants, and that is particularly of concern because, as Gil mentioned, one of the problems and adverse events noted were microhemorrhages in the brain, and these were people not taking anticoagulants. A lot of older people are taking anticoagulants. They also excluded people over 85. And in fact, a recent JAMA study from Tim Anderson, a former UCSF fellow, found that over 90% of Medicare beneficiaries with Alzheimer's disease would not have qualified to enter the Biogen study because they had at least one of the exclusion criteria. So I don't think we can say that these study results would generalize to a Medicare population because these people were younger and healthier than our Medicare population. Biogen also reported in that data that they presented to the FDA that 30% of their patients with low dose and 40% of the high dose either discontinued treatment or withdrew from the study, mostly due to adverse events or consent withdrawn according to their slides. I will say it's hard to know exactly what happened in that study because, as Gil mentioned, the data has not been published in any peer-reviewed journal, and so it's really not available for scientific review. And serious adverse events were common, as Gil already mentioned. Again, as Gil mentioned, those trials were abandoned for futility. Certainly as a journal editor, I find it disturbing that this data has still not been published in any peer-reviewed journal, and it does suggest that there are issues. Another unusual and troublesome feature is that the FDA worked with Biogen on post hoc analyses. Needless to say, we hope this does not occur routinely for the FDA to work with a company after the 2 trials that were done to show benefit were abandoned for futility and no clinical benefit. And as Gil already mentioned, ideally, a 3rd trial would have been done. The reason that FDA requires 2 trials is because we think for scientific rigor, we need to have consistent results from 2 well-done trials, and this was clearly not the results of eMERGE and ENGAGE. And again, because of the trials ending and a lot of people stopping the drug even before the trial ending, the adverse events may have been underestimated. And a House of Representatives committee and the Federal Inspector General are currently investigating these unusual aspects of the accelerated approval. So for the FDA to approve a drug without any support from its own expert advisory committee is unprecedented. So what are the implications of this very unusual approval for the Medicare program? And that's what I'm going to talk about for the remainder of my time. So I would say that this decision is a big problem for Medicare, and it's really a big problem for all of us because of the decision itself and because of the precedent it could set. So the FDA criteria for approval is that a drug should be safe and effective. And again, for an accelerated approval, that is sort of a promise based on that surrogate marker. And I will note that the FDA gave the Biogen 9 years for this post-market study that was supposed to then show clinical benefit. And as we know, 9 years is a long time for a post-market study to go. Medicare is a very complex program. I will just say that Aducanumab would be covered under the Part B. There are 2 ways, the Part B and Part D. Part D generally covers outpatient drugs. Part B covers injectable drugs that are given in a doctor's office. Part B is also affectionately known as the buy and bill program. And current Part B expenditures have been growing rapidly, mainly because of a lot of very expensive oncology drugs that are given in doctor's offices and are about $30 billion per year. So under this buy and bill, the way it works is that physicians purchase the drug, and the physicians have very little or no power to negotiate the price of the drug. The drug price is set by the company, and there is no clear relationship to value or other things between the price set by the company and the actual drug. And furthermore, physicians have no financial incentive to negotiate the price of the drug for Part B because physicians are paid the average sales price, which again is set by the company, plus 6% of the average sales price. So obviously, the higher the price of the drug, the more the physician is reimbursed for injecting the drug in their office. And this system is not thought to lead to the best value and best pricing for drugs. And there have been other suggestions for other ways to do it, which I'm going to discuss in a moment. I will say it is this system, though, that is currently operative and the one that would apply to aducanumab. And it led to this New York Times editorial from Amy Finkelstein, a health economist, that $56,000 drug blamed Medicare. And she's referring to the fact that there is no negotiation on the price of the drug, but frankly, it's not quite all Medicare's fault. That's how Congress set it up for Medicare. So Congress could, again, change the way it's done. And in fact, the Medicare Payment Advisory Commission has recommended a new and I think better technique to get rid of buy and bill and instead go to a system which actually allows negotiation. So I will say this was in 2017. I did serve on the Medicare Payment Advisory Commission in 2017. And Jay Crossan, and this is from a viewpoint that Jay Kenkovinsky, one of our UCSF geriatricians, and myself wrote in JAMA Internal Medicine recently about the aducanumab approval. Basically, what MedPAC recommended was to allow intermediary entities to negotiate prices and deliver drugs to physicians. It would maintain all of the aspects that we think are valuable about the current system. Physicians would be reimbursed for the cost of the drug. Physicians would still receive an administration fee, and physicians could potentially be eligible to share in the net savings. However, it would hopefully not allow outrageously high drug prices, particularly for drugs that have little or no clinical benefit, such as aducanumab. So currently in our system, this incredibly high-priced drug will present financial hardship for many beneficiaries and certainly for the program. Out-of-pocket coinsurance costs are estimated to be up to $11,000 annually, clearly out of many people's reach. There is much concern that it will widen health disparities. And just to point out that money that is spent on drugs without benefit is not available for services known to be of benefit. And there are services and programs that are of benefit for Alzheimer's. And actually in that New York Times editorial from Amy Finkelstein, she does mention that the cost of this drug would pay for 45 hours a week for a year of home health care for a Medicare beneficiary with Alzheimer's. The high cost of this drug would also result in increases in Part B premiums for some Medicare beneficiaries. So what are the options facing Medicare, the Centers for Medicare and Medicaid Services, or CMS? So Medicare, as I mentioned, criteria are that Medicare has to cover treatments that are reasonable and necessary. Now, one could reasonably argue that a drug where the entire advisory committee did not see a clinical benefit would not be reasonable and necessary, and that is a possibility. Medicare could choose to conduct its own evidence-based process and make a national coverage determination. So Medicare can issue national coverage determinations, which occur generally between 5 to 10 times a year. And if there is no national determination, then coverage is set at the regional level. And it means that a drug or treatment could be available in one region, but not in another. Now, Medicare has announced its intention to do a national coverage determination for aducanumab. And then finally, and this is the one I think is really important, Medicare could only cover aducanumab in the context of a clinical trial, and this would be known as coverage with evidence development. And I'll just talk for a moment about that. So the NCD would allow independent clinical experts to evaluate the evidence for efficacy and safety, and the coverage with evidence development option would allow Medicare to offer this treatment but in the context of a randomized clinical trial with the control arm being usual care. Now, obviously, as Gil mentioned, and I agree, a third trial should have been done because the first two did not show a clinical benefit. And I would say that 0.4 on a clinical scale, I believe most neurologists and geriatricians do not consider to be a clinically significant benefit. And certainly that was the vote of the advisory committee. I would suggest that the company should supply the drug for this RCT in the coverage with evidence development scenario until there is full approval with meaningful clinical endpoints. Because otherwise, by this accelerated approval, you have shifted this incredible financial burden of research from the company, which usually would be sponsoring the clinical trial, to Medicare and Medicare beneficiaries if Medicare had to pay this very high price for this drug in order to do this trial that I think should have been done before an approval. There is precedent for Medicare not covering FDA approved drugs. And for example, there is florbetapir. In 2013, MedCAC met and discussed PET imaging for Alzheimer's disease. The committee did not think that PET imaging, which showed beta amyloid protein, would lead to improved health outcomes. And again, that's consistent with what I told you earlier, that amyloid does not predict Alzheimer's disease. And so Medicare made a decision not to cover the drug and not to cover PET imaging for the detection of Alzheimer's disease. And MedCAC is the Medicare Evidence Development and Coverage Advisory Committee that does evidence reviews for Medicare. So I think this is an inflection point for Medicare. The FDA's decision to override its own panel and approve this drug may lead to permanent changes that would dramatically alter the Medicare program. And this has led to a lot of discussion about surrogate measures. This was an article from BMJ, from Jeannie Lenzer and Shannon Brownlee, which suggested that expedited approvals should remain provisional so that the drug or the device is automatically withdrawn if confirmatory trials fail to show benefit. So I'm just going to spend a moment on the estimated cost to Medicare and then wrap up. So it's hard to predict exactly how many Medicare members would get this drug or would request this drug. But if you estimate something between 15 to 20% of Medicare members, and remember this was approved for all Alzheimer's disease, although then the FDA sort of walked that back and said, no, they would just approve it for early Alzheimer's disease. Although, as we know, once a drug is approved, it can be prescribed off-label for any patient. So if we take this conservative estimate of 1 million of the almost 6 million Medicare members who have Alzheimer's disease, the cost would be $57 billion a year. Remember, all of current Part B drug spending for all Part B drugs currently is $30 billion a year. So we are talking big money. So I think either Medicare Part B expenditures are on a path to skyrocket in the future, or Congress needs to consider and enact legislation to change the way we set prices for prescription drugs. And either way, FDA approvals based on non-validated surrogate measures should remain rare and well justified with validated, so that we can have confidence of clinical benefit for patients. So I think this approval represents a threat and an opportunity. And again, I think it's really important to show net clinical benefit. It's time to control drug costs, both to protect Medicare beneficiaries and only offer safe and effective drugs, and to safeguard the financial solvency of Medicare for all. Thank you.
Fantastic. Thank you, Rita. Thank you, Gil. Great discussion. We have tons of questions. Let's see how many we can get through in the next 15 minutes. Rita, we're both editors of journals, but I'm not on a panel and you are, so I get a chance to ask you and I don't have to answer. One of our folks said, listen, why hasn't this been published? 3 years have gone by since these trials were done. Gil had to walk us through data that he grabbed off of publicly available sites. Speculate, Editor Redberg, why hasn't this been published yet? And is that something concerning or should be required before the FDA and others rule on a drug like this?
Sure. Happy to speculate. And I will say, as you may know, I'm sure you do, Axios did cover and suggest that this article was withdrawn by the company from review at JAMA. But certainly, in my own experience in editor, I'm sure is yours, you know, when particularly a sponsor has a particular way they see the data or the way they would like to spin the data, and they cannot get eye to eye with the editor or the editors and the reviewers. I mean, it's you know, it's really important when you look at the data and it's a negative trial, we need to report that negative trial. And if the sponsor, so I think it's very possible that the sponsor and the editors could not come to an agreement on how to portray those results in the conclusion section based on the results of the trial.
Gil, I've got two questions for you. I'm sort of melding a couple together. Let's first sort of understand just a little more on how accurate this marker is. So it sounds like people were selected if they had a biomarker on a PET scan that said that they had amyloid. Are there people with Alzheimer's who don't have that? How accurate is that for that we're treating people who actually have Alzheimer's disease? And then I'll give you the follow-up.
Yeah, thank you, Andy. So I think, you know, there's a lot of confusion about terminology and what Alzheimer's disease means. I think that for many years, and certainly when I was in medical school, we were taught that Alzheimer's disease is a clinical syndrome, which is, you know, related to progressive changes in memory and other cognitive domains that leads to functional impairment, and that you've ruled out, you know, metabolic, structural lesions in the brain, et cetera. Neuropathologically, since Alzheimer's first case report, Alzheimer's is defined by plaques and tangles. And so by definition, a neuropathologist would never say that someone has Alzheimer's disease unless they have both plaques and tangles. And in fact, they'll comment on the degree of and the distribution of plaques and tangles. And until we had biomarkers, we were really stuck, I think, with the clinical definition. But with the biomarkers that we have, CSF measures of A-beta and tau, PET scans that are very costly and not accessible, but actually very highly accurate, and I'll get to that in a moment, and increasingly plasma tests that are looking very promising, and I think will greatly increase access and cost effectiveness of biomarker testing. The definition of the disease is shifting towards that biological definition, just like a neuropathologist would diagnose a brain with Alzheimer's if they found plaques and tangles. Similarly, we with biomarkers can determine whether amyloid and tau is present and define, that is how the disease is defined. So I think, you know, dementia can be caused by Alzheimer's. It can be caused by other diseases, vascular diseases, Lewy bodies, other protein aggregates. So let me make an analogy in medicine. You know, someone can get dementia from different causes, just like someone can get heart disease from different causes. One of the major causes of heart disease is hypertension. One of the major causes of dementia is Alzheimer's disease, defined as plaques and tangles. And you can certainly have dementia with, high blood pressure. You can certainly have, I'm getting confused by my own analogies. You can certainly have cardiac disease without hypertension, but you cannot have hypertension without elevated blood pressure. Similarly, you cannot have Alzheimer's disease if there's no amyloid in the brain. So just the definition is important.
So let's pick up on that based on, I think, a really important point that Rita was making at the end, and then a question, which is, so how many people is this really going to involve, Gil? So you know, there are people who are given a diagnosis of Alzheimer's disease, but do not have the kind of accurate diagnosis you're describing with these quite expensive at this point tests. So does that mean that if we were to use this drug, we should use those expensive tests to find the people who really have AD? And if that's the case, how many people are we talking about that would be eligible for this drug if we just use the trial criteria?
Yeah, no, I think these are very important questions. Fortunately, there are very accurate CSF biomarkers now for amyloid and tau that can determine whether someone has this pathology in the brain. And that certainly is more widely accessible and much more cost-effective than PET scans. So absolutely, I do not think anyone should be considered for treatment with this drug unless there's evidence of amyloid in their brain. It would not make sense to do that. Similarly, I think that based on what we understand of the biology and evolution of the disease, it would not make sense to treat people who have advanced dementia. They were not included in the trials. We don't know about safety or efficacy in those individuals. And I think that, this is really the very earliest days of precision medicine approaches in Alzheimer's disease. And I think that just as this has progressed in other fields of medicine, we will eventually identify people who will respond to different treatments based on clinical factors, based on genetic factors, based on biomarker profiles. And this is just the beginning of that era. And so I think that if this treatment is used at all, it should be used in a very select and small group of people who would be similar to those individuals in the clinical trials, where we can at least as clinicians talk about the ambiguity of efficacy and the risk of ARIA in some context. And this, treatment, if it is used, should really be used by experts who can understand the nuances of the data and can engage in shared decision-making with patients about potential risks, benefits, and honestly, with this drug, a lot of unknowns, especially around clinical benefit.
Rita, let me ask you a question that's come up. So, this is, as you said, it could be incredibly expensive, even if we were to narrowly define it as Gil was suggesting. Does the FDA even think about, when it's trying to make these decisions, expense of a drug? Should there be a different threshold for approval based on some of these more societal implications for cost? Is that ever done? Should it be done? Or is that the FDA just approves everything and it's CMS's role to think about that? Where does cost come into these types of decisions, Rita, in the United States, or should it?
Well, that's a great question, Andy. And one of the reasons we have a $4 trillion healthcare system, we spend way more, twice as much as the next highest spending country in the world, yet we don't cover all Americans with health insurance, very spotty coverage, it is because we never talk about cost. We pretend that we have unlimited money to spend on healthcare. And in fact, when I started, you mentioned the London School of Economics and Health Policy, we were at 9% of GDP for healthcare. We're now going to break 20% of GDP for healthcare. And we still don't have the best statistics. People aren't living longer, they're not living better. And so I think when we're looking at really expensive drugs, we want to help people to live better, and we want to help people to live longer. The FDA does not consider cost, and Medicare does not consider cost. So we all pretend that costs are not an issue and we have unlimited, money to spend on healthcare. But frankly, for this drug, before you get to cost, you have to think about, is there clinical benefit? This drug has not, the entire advisory committee, nobody found clinical benefit for these drugs. So I think, that kind of made it a little easier. Why even talk about cost when we don't have clinical benefit? And the last thing I just wanted to add is, if I have to have a test to find out I have a disease, I just won't take the test, because if I don't have it clinically, I think we're taking a lot of people and who hasn't forgotten their keys somewhere, left their phone? I've done that. If you don't clinically have dementia, I don't think I want to take a test and then be told I have something.
Yeah, let me pick up on that. I was going to ask a different question, but let me throw that to Gil. So Gil, I think what you're suggesting is that, many of these Alzheimer's treatments, they are going to be too late if we wait till someone has big symptoms. And so rather, it sounds like the future of the field is kind of thinking what Rita was just saying, that you're going to have to screen people at a pre-symptomatic age, maybe at the time they get their colonoscopy, and then these drugs are going to be more effective. Is that kind of where the field is going, Gil?
I actually think that it is headed in that direction. you know, I think maybe I have a little different perspective, but, you know, I want to know my blood pressure, even if I'm not having symptoms of, you know, malignant hypertension. I want to know my fasting blood sugar, even if I don't have symptoms of diabetes. I want to know my lipid panel, you know, even if I don't have heart disease or stroke. And so I think that, you know, more and more we're thinking about these pathologies as risk factors. And there is a well-established literature that shows that having amyloid and tau in your brain is a significant risk factor for developing cognitive decline in the future. Not everyone who has the proteins will develop cognitive decline, just like not everyone who has vascular risk factors will develop MIs or strokes, but these are significant risk factors. And so I think that ultimately we are thinking about addressing these risk factors proactively. There are also a lot of lifestyle things that we can actually do to try to reduce our risk of developing dementia. And these should not be understated because they actually are well validated in studies ranging from animal models to large randomized trials, things like exercise, healthy diet, sleep. So these have actually made a tremendous impact. And we think that better treatment of vascular risk factors has reduced the incidence of dementia in many countries, even if not the prevalence, because the population is aging. And so the prevalence is increasing even as the incidence is decreasing. And so I do think that's the future. And I think that plasma markers are going to really make that possible because they will be accessible and they will be cost effective. They may need to be followed ultimately by something like a lumbar puncture, but at least when you're thinking about surveillance, the plasma markers of amyloid and tau are extremely exciting and I think game changers in our ability to modify the course of the disease.
Rita, we have like 1 minute left, but let me hit one in your wheelhouse. Gil showed us the results of these trials, which were extraordinarily homogeneous in terms of their racial makeup. The FDA approving drugs without trials that are diverse in terms of both sex and race and ethnic background. Where are we on this?
Well, as Gil just showed us, unfortunately, I don't think we're where we need to be. And I mean, I've worked on this for 20 years. I mean, Sanket Dhruva, who's now on our faculty, when he was a first-year med student, we looked at clinical trials that were reviewed by Medicare for Medicare coverage decisions and found that they were much more white, much more male, much younger, much healthier than Medicare beneficiaries. I've been invited to FDA to talk about why there aren't sufficient women in clinical trials for drugs and devices. I said, you know, it's very simple. I mean, they have recommendations for gender and racial and ethnic diversity, but they're having big letters across the recommendation. These are not requirements. These are guidelines. These are not binding. Well, if you can get your drug approved without, you know, having a gender and racial and ethnically diverse population, and for some reason sponsors see that it's cheaper and faster, then they're going to do it. I said to the FDA reviewer, I said, it's very simple. Just reject the application if it doesn't meet your guidelines for gender and racial and ethnic diversity. And they said to me literally, well, that would upset the industry sponsors. I said, well, I think the FDA is there to serve public health, not to serve the industry sponsor, but I'm not sure what the FDA thinks its mission is these days. It's of great concern.
We're going to leave it there.